Hi guys, so we're going to look at some genetic questions now, and there's a few in a row here which hopefully should really hone our technique. Before we do this, I would just like us to think about the definitions that we need to know for this part of the course, and we do need to know them, okay? We need to know that an allele is a version of a gene. We need to know that homozygous is two alleles that are the same of a gene, so that could be big B, or small b. That heterozygous means that you've got different alleles at a gene, so that could be big B, small b, for example, if B. Remember, you might have questions in which they use a different letter, like there's one later, I think, where they're using R, so it could be big R, small r, for example. Okay? So, our definitions carrying on are dominant and recessive. Now, these have got very specific definitions and we need to be sure of them. Dominant is where an allele will be expressed in the phenotype, which we'll come on to in a second, in either the homozygous or the heterozygous condition. Can you see, because big B is dominant here, it obviously is going to be expressed with big B, but when there is a, inverted commas, choice between big B and small b, if big B is expressed in the phenotype when there's a small b there, then it's clearly dominant. And recessive is an allele which is only expressed in the double recessive condition. So the word here is either for dominant, and the word for recessive is only. And it's only expressed if you've got double recessive. As soon as you've got a big B in there, it is not expressed. We talked about phenotype. Phenotype is the characteristics you see, the characteristics expressed, okay? Excusez-moi. Characteristics expressed. The genotype is the two alleles found at a gene, just written. So it could be big B, it could be big B, okay? So the genotype, the two alleles together is a genotype, okay? And we have, those are the definitions that we need to know, okay? So if I've forgotten any, I'll put them in as we go. So that is a background to the kinds of things that we need to be thinking of and to know here. Now, the other bit of background that I just want to do is that remembering that what we have whenever we have sexual reproduction is we have a sperm, for example, in us, a sperm and an egg that fuse together to form a zygote in us of 46 chromosomes, okay? I'm using us as an example because it's much easier for us maybe to identify with this, but of course this is the same for any organism, okay? Now, though that sperm came from a father, a male, and the egg came from a mother, a female, and we actually write that down as those. And so the process by which we got that sperm and egg in us is meiosis, okay? And so we produce gametes, and then those gametes fuse, and the fusion of those gametes will create a new, and I used this in the last video, diploid. So these are haploid, interesting word, and diploid. Okay, so the fusion of two gametes will form a diploid zygote. Okay, so that is what happens in sexual reproduction. Now, the other thing I wanted to just explain here is that when these 23 come together, you've got 23, I'm just going to pretend this is 23 chromosomes from dad, from the male, and 23 chromosomes, pretend that's 23, from the female. Okay, and what they do is they pair up. And so I'm going to look for some similar ones here. So you see that one there, pairs up, for example, with that one there. And they would pair up, and they would have alleles on them which are versions of a gene. So if this was eye colour, for example, in a human, the father might be saying big B and the mother might be saying small b. Okay? And those alleles would be in the zygote. And the actual expression of the phenotype, remember what we see, the characteristics, would be as a result of those two alleles lining up with each other, one from the male, one from the female, and there being, depending upon which one was dominant and recessive, we know that eye colour, big B is dominant, will be expressed in the phenotype. So those two chromosomes would line up together, and then looks for something else. That one and that one would line up, and that one and that one would line up. Okay, and so you get one version from the male and one version from the female. So that is a background to the process of sexual reproduction and how the characteristics come out. One allele is denoted by the male on a certain gene, and one is denoted by the female. Okay. Now, as I describe all this, if this isn't clear or you're unsure of this, then you need to make a note of that, and we need to get together for a one-to-one. Okay. So that is how sexual reproduction happens as a background, and we can write the probabilities of certain phenotypes, expressions of alleles, with genetic diagrams. And this is what this question's about. So with that background in mind, okay, let's go on to these questions. For many years, scientists studied the organisms in an area of grassland. So we're talking about plants, we're not talking about humans. One of the animals was a species of black fly. Sorry, apologies. It's in the grassland, but the animal we were speaking was a black fly. In this population, only one allele, B, existed for colour. So one allele for colour would mean that all of the flies were big B, big B. There were only big B flies. Okay? A mutation occurred which produced a new recessive allele, small b, which could produce a green colour. Excuse me. So a mutation occurred where suddenly there was a small b. And the small b could produce a green colour. Interesting. But if the small b is recessive, then the only way you're going to get a green colour is if you've got double recessive. Because remember, dominant, if it's in the heterozygous condition or the homozygous condition, it's going to come out as black. So that's our thinking. Let's see what this question will ask us to do. Draw two genetic diagrams to show how the single B allele in just one fly was able to produce homozygous, double recessive, small b, green flies in 2 generations. Okay, so this is actually very straightforward. All the black flies were big B, but then one day there was a mutation in which we got a small b. And these two flies would have mated together. Now, the gametes that they could have produced in their sperm and egg are the big B here could have been donated, or the big B there could have been donated. Let's say that this is the father, for example, and this is the mother. The mother could have donated her big B as her version, or she could have donated her small b. Okay. Now we don't know which of these happened, and we don't know which of the sperm and the eggs mixed. So the way we can do this is to produce a Punnett square. And a Punnett square is we take the two genotypes, gametes, and so for the male we've got big B, and for the female we've got big B, small b, and then we multiply them out. So we get big B by doing that one and that one. That one and that one produces big B, small b. That one and that one produces big B, and that one produces, I do apologise, sorry, that one, good, and then that one. Okay, so we've actually got a 50-50 split on homozygous dominant and heterozygous. Okay, now that's the first generation. Apologies, I'm having to rearrange this. So that's the first generation. Okay, now our second generation, we're looking to see, oh my goodness, this is very irritating. Okay, do not touch this button, Mr. Abraham. Don't touch that button. Right, so we are looking to try and show how double recessive green flies could be produced. Well, we have got here these two homozygous flies from the first generation. If we were to mate those together, the gametes that this one can produce are big B or small b. The gametes this one can produce are big B, small b. And if we put those in a Punnett square, we get the following. Big B, big B, big B, small b, big B, small b. So that we put the capital letter first, and then small b, small b. Aha! There it is. There is the small b that would lead to a homozygous recessive, which equals green. These three, that's going to be black, that's going to be black, and that's going to be black. Okay, and going back, because these have all got big B's in, that's going to be black, that's going to be black, that's going to be black, and that's going to be black. So going back, we didn't have any greens in the first generation, but by crossing to heterozygous offspring together, we get one small b, which in this case is green, and therefore we've proved it. Okay, now moving on. Although this new allele was recessive and the mutation only occurred once, a large proportion of the fly population was soon green, suggesting in terms of natural selection why the recessive B allele was able to spread through the population. Now, I'm interested to see how many marks this is. So this is 3 marks, okay? And so with that in mind, we need to put 3 good points in. And this is about natural selection and our exact process. We've already seen what the mutation is in the formation of the small b. And so if you think about our sequence of natural selection, we've got 1, you have a mutation, 2, which gives an advantageous allele, which 3 increases the chance of survival, 4, increased chance of reproduction, 5, offspring have allele, 6, over time population increases with advantageous phenotype due to survival and death of others. okay, not having. Okay, so that's our whole process. So whenever we get an evolution question or a natural selection question, this is our process. And now we just need to think for 3 marks, which is the relevance here? Well, we've already done the alleles, okay? So the mutation has gone, but we can say that there's an advantageous allele, which actually we should say goes to the phenotype. So there's an advantageous phenotype, which is the green, which is camouflaged, okay, which would mean more chance of survival due to less chance of being eaten by predators. We're filling in the gaps here. And that means that the allele will be passed on and the offspring have it and survive. So in this question, we're thinking, there's all my natural selection points. I can fit them in here. I don't need the mutation part. I can actually just add them from then, okay? So you can see on the mark scheme here, by the way, the mark scheme has talked about the going through what I did before. So the mark scheme here says green colour gives an advantage, camouflage, more green flies survive to breed, pass on their genes to the next generation. I prefer allele, okay? And then we've talked about the offspring surviving, which I think we should do just to double check on the marks. Okay, so that is our question there. So there is our question 5.